So there's still almost as much, I mean, it's a factor of maybe 2 or 3 variation over the cycle. Here it's a variation of 10. And the reason is, as I explained, in the winter, this is oxidation of iron sulfide. There is no storage for ammonia, and therefore the ammonia flux goes down to nothing. Once you've burned out much mineralization shuts off. There's still G2 down there, but the mineralization rate has gone down because the theta is so high. We're not talking about silica. At R64, which becomes anaerobic in the summer, you can see from the SOD, it's shutting down because there isn't any oxygen. And so, of course, you can't get any sediment oxygen demand if you haven't got any oxygen in the overlying water, so the model is turning it off. It's a lot more ragged because the aerobic layer is shrinking dramatically. Ammonia fluxes are doing, nice regular ammonia fluxes. There's nothing. And here's the winter, you see. And finally, remarkably, the phosphorus flux in this model really worked better than it should. In most of the models we build them so that they behave in a way that we think they ought to behave. This more or less came out of it. It just works. This is, by the way, the back diffusion of the high overlying water phosphate after the big release. Once the aerobic layer forms, the partition coefficient in the aerobic layer takes hold and it all sorbs. It gets transferred out of the overlying water into the sediment, and that's why there's a slight negative. One other interesting set of data. Know what those are?
Merle tanks.
Good, Merle tanks. Yes. Those of us that are old enough know what they are. The University of Rhode Island built a series of tanks, 10 meters high, I think a couple of meters in diameter, 6 meters high, 6 yards high, a meter across. And Narragansett Bay water was pumped into these tanks. They built a special set of coring devices that were shaped like pie-shaped devices. They would go out in the Narragansett Bay and pull up sections of intact core and put it into the bottom of the Merle tank. They had a plunger that would scrape up and down to keep the periphyton from building up and also to mix it. So they wouldn't let it stratify. You could let it stratify, but that complicated things. So these things were well mixed. They had kept the temperature constant, and they varied the nutrient concentrations. And there were, I forget how many there were, but that's what it looked like. They're still there. Here's how they decided the loading. Scott Nixon was the brains behind this, and Candace Oviatt, also she. And Nixon made estimates of the aerial loading rate to the estuaries in milligrams per meter squared per day. I don't know how he did that. I mean, what is the aerial loading rate to Massachusetts Bay? Well, how big is the bay? I mean, but anyway, it's a log scale, so I wouldn't get too upset about it. But what's interesting is where he set the loading rate for Merle. So the 1x was sort of his guess for what Narragansett Bay looked like. San Francisco Bay was here. These are the Merle tank loadings. You see it was logarithmic, very intelligent. 2, 4, 8, 16, New York Harbor, New York Bay, 32. So in order, variation over an order of magnitude or more. And here's the sediment flux model compared to the data. Here is the control. This is ammonia flux. You see it increasing. You see these negative things? That's the overlying water pumping ammonia into the system. We could never reproduce that. We didn't have any idea what that was about. And you can see at the highly loaded systems, things are getting pretty ragged. But in through here, it was working okay. Nitrate fluxes. See the denitrification? A lot more denitrification than we actually could calculate. Jeremy Testa's correction. We never reran this, but it'd be interesting to do that. But here's SOD. You see how constant it is? Look. Very little variation. Look at the variation in ammonia fluxes. 0 to 100. 1 to 3. damped out because you're just forming all of this iron sulfide that is sitting there just slowly being phosphate fluxes.
What's the x-axis?
Oh, I'm sorry. Yeah, good question. 3 years. 1981, 1982, 1983.
Thank you.
Of the control tank, I'm sorry. 1x, 2x, 4x, and so on. So this is 3 years worth of data. Silica flux, Damien figured out that we didn't have a temperature correction on the silica solubility. That fixes that. So that's it. I'm not going to show you any more. So, I hope what I tried to do was to give you somewhat of a historical picture on how these things were developed, but hopefully to build up your intuition so that when you see this stuff coming out, you can think, what are the 3 or 4 important things? So I remind you the 3 or 4 important things are that, especially for SOD, it is a combination of 3 reactions you have to worry about, which is, and you don't worry at all about the aerobic oxidation of organic matter. It simply doesn't amount to anything. It is the aerobic oxidation of all of the reaction products. And the 2 big ones are iron sulfide and hydrogen sulfide and methane. Those are the 3 things that track. And the way we get SOD, to go all the way back, is we just add up how much oxygen is consumed by each of these reactions. Just add it up. We know the rates for each one of them. We add it all up, top and bottom, well, in the aerobic layer, of course, and that's SOD, and then that feeds back into S. So the model has an iterative loop built into it. I forget what we do. We guess the SOD, and then we calculate.
Well, it's a binary search.
It's a binary search?
Minimum, maximum, and binary.
Right, we just hone in to get the equality. So S has to match this sum. Okay?
You have the CH4 in H4.
Oh, I should have methane in here as well.
Okay.
Well, ammonia, I should have methane there as well. I didn't put it in. You're right, there should be methane. Yeah.
Just to make sure I understand.
Sure.
Thyroid perturbation in the PO4 model and for the sulfate as well, because the sediments are actually moving, but the sediments are actually moving for the NO3 and NO4, and that is moving the surrounding the inner capillary water, or whatever you want to call it, but that's captured in the diffusion rate.
Correct. It turns out that we looked at bioirrigation, which is the fact that organisms are breathing, and as a consequence, they're breathing water down at the anaerobic layer, and then they're excreting it in the aerobic layer. So there's a transfer called bioirrigation. It doesn't amount to much. Turns out that this transfer is bigger.
And those transfer rates will do it for the nitrogen.
Yeah.
Okay.
Yeah, the mixing of, there's not enough particulate ammonia and nitrate to amount to anything.
Right. So the diffusion coefficient is sufficient to capture the nitrogen.
That is correct. That is correct. Okay? Yeah.
First, thank you for your lecture, so much in depth, we learned so much.
Good.
And I just have a quick, practical question.
Yes.
I think anyone, Carl or Jim, you can help. Is that I kind of found that the diagenesis model kind of relatively easy to be followed up.
Easy to be?
Just follow up.
Blow up?
Yeah.
Oh.
And it essentially happened in the function we call the Zebran, or kind of you call it
Zebran.
Yeah, exactly.
Yeah, it's the root finder.
2 things. Yeah, we've had the same problem.
Yeah.
Usually, at least in our shop, most of the time it blows up is because somebody made a mistake in putting in a huge load. Maybe they slipped a decimal point or 2 or 3, and you're putting down so much diagenic material.
Right.
It
The
blows.
aerobic layer shrinks to nothing, and the whole thing just blows
And
up.
the other thing is, you have and I assume you have the same thing, Carl, that you try not to calculate negative concentration, so you have that. So in principle, oxygen can get really, really, really small, and when it goes to try and do the mood solver, sometimes you have problems. So what we ended up doing is setting like a minimum oxygen concentration, even if the algorithm tries to go through it, we set it to like 0.01 milligrams per liter. And if you do that, that should take care of most of your problems if that happens. So two things, it's either you've got an input error, or that the minimum oxygen concentration gets so low that that Z-Brent root finder has a problem. So if you put a floor on your oxygen, normally you might say 0.001, what's the difference between 0.001 and 0.01? So we set it a little bit higher, and that sort of takes care of, should take care of your problem.
Thanks.
If you specify a minimum concentration anywhere.
We quit looking, you know, a lot of this is old members had to come back, but we were always getting, well, when we get a Z-Brent failure, what we really found is we were putting garbage into the sediment model. And I don't mean physically garbage, I mean we were feeding it nonsense numbers.
Right.
And as a consequence, that algorithm is the first place it blows up.
Yeah.
So, I mean, when we see a Z-Brent failure, we don't look at the sediment model anymore. We just say, we've got some nonsensical numbers somewhere. And we'll look at, for example, the Bay model, and we'll see there's negative ammonium concentrations because we have an instability. Or that thing I suggested, you know, like about the volumes, that if your volumes don't add up, they go to zero. So inevitably, if we get a Z-Brent failure, it isn't really the sediment model, it's garbage in, garbage out. So don't even look at the sediment model, look
Yeah, but if you
Look elsewhere.
So two places it said 90% of our problems would be somebody made an error in the input deck, loads or whatever. But the other problem is even if those loads check out, if you do have a heavily organically loaded system where the oxygen gets very low, just set your threshold value so that if it tried to go below 0.01, you'd set that, or we just automatically set our lower bound for oxygen in the sediment model to 0.01. And the way you can do that is you just put a statement in, because one of the things that the sediment flux model does, it takes the concentration of the overlying water column and puts that in as O2O. And we just say we put a max of O2O to the overlying water column of 0.01, so that way it never gets below 0.01. But I am going to ask a quick question.
Sure.
Not so much for Dom, but maybe what's going to be covered in the next day or two. Are you going to cover why we went to now a reactive G3, basically the kind of window problem, or not?
No.
You're not?
It's a little,
Well, is it worth verbally describing why that was done? Because 99.99% of the time, you'll never worry about a G3 reaction.
Yeah, exactly.
So it'd be worth perhaps describing that, and if you want, I can do that at another time, or, you know, we can talk about it maybe tonight.
That's interesting.
It's covered.
Yeah, I didn't realize, I was wondering why you did that, and the answer is you needed an aside from neatness.
Well, it really had to do with Connor Windows, really the first project anybody's worried about resuspension of organic material that they've been sitting there for 5 or 10 years.
Got it. I see.
we'll talk about, we'll
Yeah,
make it a
talk about
point
that, okay.
to talk about that.
Yeah.
We also saw some negative ammonia flux in Delaware River, and we couldn't figure out. we were thinking, okay, possibly due to high input of ammonia.
It's the only thing it can be.
Yeah, which did happen. You know, some industrial or wastewater treatment plant discharge. But we were thinking, like, you know, that's if the flux is from water column to sediment, it shouldn't be just a short cut. Like a short time period, because the pore water is like the
Well, it
volume
depends
is
if you
low.
have enough oxygen in your overlying water column and you're getting the nitrification, so that ammonia is disappearing. I would agree, one would think if you put in that much ammonia, the
but
water column would go to
because
zero.
at the same time we are having a high positive ammonia, so it's like you know, ammonia is releasing, but sometimes it's going.
Yeah, we never could see all those negative ammonia fluxes. We could never figure out where they were coming from. Maybe, I don't know, I mean, you know, this thing doesn't know about Aminox, the new, you know, that reaction.
That nitrate goes back to ammonium.
Right.
So when you denitrify, you know, maybe if you're denitrifying in the overlying water or just near the, I don't know, could be.
Yeah, take a look at some of the work by Ann Giblin. She's one of the ones that's been looking at anomops and tried to figure out whether it is something.
Does it amount to anything?
It may depend upon where it occurs, and I think it may occur more in sandy sediments, if I remember some of Ann's work.
Yeah, I forget. But that had us puzzled.
You want to check,
Bacteria.
you want to check too, is light hitting the bottom?
Yes,
Oh, right.
right.
If light hits
Yeah,
the
that could
bottom,
be.
the algae that sit on the sediment-water interface will intercept and if all you're measuring is the flux, you'll get, the action of algae and light will impact that flux tremendously. So if it's really shallow water, you've got to ponder that.
Yeah, that's a good point. That's a good point. Yep.
Yeah, because some of, what you have it in your benthic algal model, if it's shallow enough,
Sure.
and what's interesting is actually doing the calculation of what it would be in the light versus the dark, and then the total.
It's dramatic.
The difference.
Yeah.
And that affects SOD, because you can actually have a negative SOD if it's producing enough oxygen due to benthic algae.
So here it is. Go buy yourself a copy. I don't make any money on this. And there are now used versions available. And you can
You
see,
do a book signing.
yeah, you can see what's in it. There you go. Yes.
Well, somebody's got to do it. Thank you.